So this is going through the nervous system questions, that the last three questions in your component 3A2 book. So I've got my homework kit with me: pen, pencil, highlighter, ruler, calculator, because you'll need all of those things in the exam, and you should have them while you're doing your homework too. So this is a pretty typical question that you might come across in the exam. It starts with a big sort of pile of information. They've got a graph, so it's covering a bit of sort of the numeracy aspects, and this one particular has a lovely bit on the core at the beginning. So and a little bit of synoptic from module 2. So we need to read the information. So it tells you that botulinum toxin is a poison produced by the bacterium Clostridium botulinum. It's involved in a poisonous condition called botulism, associated with spoiled canned food. And then we get into the real nitty-gritty. So botulinum toxin causes muscular paralysis. So you should already be thinking, oh, hang on a minute, that means that we can't contract the muscle, we're not getting depolarization. And it can be used as an injectable medication for the treatment of facial wrinkles, Botox treatment. Now, why you would want to put some poison in your face is anybody's guess. The toxin, so what are they going to tell us about it? The toxin is a single polypeptide, and its primary structure is non-toxic. Tertiary structure becomes a powerful blocker of synaptic transmission, so that should now alert you to which sort of bit of the nervous system booklet you should be in. This toxin acts as an endopeptidase, so it's now going to tell us how it works, which attacks a protein in the membrane of the synaptic vesicle. And what does that do? It prevents them fusing. So now I'm thinking, oh, hang on a minute, if it can't fuse with the presynaptic membrane, it can't actually then release any neurotransmitter. So that's things that you should already be thinking of just as you're starting the question. So we start off, what is meant by the following terms? Straightforward definitions. Primary structure, tertiary structure, endopeptidase. So primary structure, straight out of your brain onto the paper. It's the sequence of amino acids. And I'm just going to finish that sentence to ensure that the examiner knows that I know what I'm talking about in the polypeptide. Tertiary structure, again, straight out of your definitions. This is the further folding of the polypeptide into a 3D shape held in place by R group interactions. And if you had time, you could list a few, like ionic disulfide bridges. Endopeptidase, so this is a synoptic one from component 3. So endopeptidases, these are the ones that break peptide bonds in the middle. So endo means inside, doesn't it? In the middle of the polypeptide. So, part B, so we've dealt with the synoptic stuff and the core. Describe. So this is sort of AO1 stuff. the events which normally occur between the arrival of an action potential, so we're starting after that point, and its transmission across a neuromuscular junction. So this is synaptic transmission. So there's quite a lot we can say. So the action potential arrives, and we've got calcium ion channels open, and calcium ions enter the synaptic knob. They cause, remember what they are involved with, so they cause synaptic vesicles. That's what they were called up here at the stem of the question, synaptic vesicles, to migrate to the presynaptic membrane. This is definitely an explanation that you need to memorise. It's one of those stories. You can tape it, listen to it again and again and again, and write it down again and again and again, but you've got to get this one into your heads. So, cause presynaptic vesicles to migrate to the presynaptic membrane. Now this is key. The vesicles fuse with the membrane. They don't just bind to it, they fuse with it. So you do need to remember to refer to fusion and release a neurotransmitter into the synaptic cleft. And this is always worth putting, by exocytosis. Exocytosis is very often worth a mark just on its own if you're talking about exocytosis of the neurotransmitter. The neurotransmitter then has to get to the postsynaptic membrane, so it diffuses across the synaptic cleft. And this is the bit that nobody ever gets wrong, and attaches, and this is a place where you could use binds, to receptor sites in the postsynaptic membrane. And it's that that causes sodium ion channels to open and depolarize. Now, what are we depolarizing? We're at a neuro, so we've got a neuron muscular junction, and depolarize the muscle cell. Okay, phew. So we're still on the subject of synapses. So we've described what normally happens, but we've now got parts 2 and 3. So this is all going to be about synapses. So when they change a letter, and they've stopped doing the dotty things, and they change the letter, it's going to be a change, a slight change, a shifting sort of topic, if you like. So, explain. So we need to give reasons now. Explain the effect you would expect botulinum toxin to have on the action potential. So we go back to the beginning. What do we know about botulinum toxin? The toxin acts as an endopeptidase which attacks the protein in the membrane of the synaptic vesicles, preventing them fusing with the presynaptic membrane. So normally we've got this fusion and exocytosis. So this is going to prevent the exocytosis. So the toxin prevents fusion of the synaptic vesicles so neurotransmitter, can't spell it, neurotransmitter is not released into the synaptic cleft. It doesn't bind to the receptors, so no depolarization can occur. Excellent. So, more about synapses and Botox. The effect of Botox treatment can wear off over weeks to months. Suggest, so this is one of our AO3, what leads to this loss of effect? So now we have to think, well, what might make this scenario happen again? Well, obviously we have to get rid of the toxin. So you could suggest that the toxin breaks down. Some people suggested lysosomes might break the toxin down, so it doesn't have an effect. You might suggest that new vesicles are made. You might suggest that more synapses are made. So it has to be something sensible about the toxin being perhaps broken down, destroyed by the immune system. It has to be something sensible, but you're not expected to know the right answer. Actually, while I was marking this last night, I thought, I wonder how that really does happen. And it really does happen because more synapses are made, and therefore the muscles can contract again. So we're going to shift again, because we've got part C now. C means we've shifted the topic. Tetrodotoxin, never heard of it. Poison, which affects the transmission of the nerve impulse. Okay, in a different way. It affects the transmission of the nerve impulse. We've got oscilloscope traces. So this is an untreated axon. So remember, we need to look at what these things are showing. You need to read the whole thing. So we've got the normal, it's going from -60, +40, hyperpolarizing, back to resting, milliseconds, millivolts. And then here we've got oscilloscope trace from an axon treated with this poison, and it's just here, isn't it, not doing anything. We're not getting that peak. So this is staying at resting. So, thinking to what we know is that that is caused by sodium ion channels opening to cause depolarization. And this is the repolarization, hyperpolarization bit. So I'm just going to go and remind myself it's potassium ions and hyper. But the key thing is that this is just staying at resting potential, so it's not actually doing this bit, is it? So let's get to the question then. State the effect of the toxin. Suggest how it could work. So the effect of the toxin is it prevents the action potential. depolarization, and therefore the action potential. So I've got both of those terms in. I'm not sure because I'm not a mind reader what the examiner wants, and therefore the action potential. In the axon. Now what could stop that? Well, we've got to be stopping these sodium ion channels opening. This could be, as you're suggesting, because explanation, the sodium ion channels are blocked or prevented from opening. Key thing is we're not opening them up. And again, you're not expected to know how any of these toxins work or even have heard of them. I can't remember whether tetrodotoxin, it might be pufferfish, that one. I think the dendro one's the mamba. Okay, that's a question.